Now we are ready to talk about nucleotides and nucleic acids. So a nucleotide is the monomer of a nucleic acid. And so we'll talk about what that means in a minute. But generally, the word nucleic acid just refers to the fact that these are molecules that originally were isolated from the nucleus, or can be purified from the nucleus of eukaryote cells anyway. And they act as acids in acid-base reactions. So that's all that means. But they consist of these subunits called nucleotides. So a nucleotide consists of one of usually four nitrogenous bases. Actually, there can be more than four, but within any given nucleic acid, there are only four different bases that you can have, and that is attached to a pentose sugar called ribose. So we learned about carbohydrates in the last chapter. So the sugar group that is part of the nucleotide is called ribose. Now there are at least two different versions of this ribose. If it's a standard ribose molecule, there's a hydroxyl group at each carbon, or a typical ribose has a hydroxyl group at each carbon. But in nucleic acids, there are basically two types of nucleic acids defined by whether or not there is an oxygen group or hydroxyl group at the 2' carbon of the ribose. So if there's an oxygen there, we just call it ribose. If there's not an oxygen there, we call it deoxyribose. So when we talk about ribonucleic acid or RNA, we're talking about that kind of ribose. Whereas if it's DNA, that's deoxyribonucleic acid. And then a phosphate group attached to the 5' carbon of the ribose makes it a complete nucleotide. You can have a nucleotide without the phosphate, and that's just called a nucleoside. But the total group together, all three, the base plus the ribose plus the phosphate, we call a nucleotide. And so as we'll learn, connecting these together forms a nucleic acid. So again, ribose is a 5' carbon sugar, and it is so that makes it an aldopentose. So in its linear form, it has the aldehyde group at the end. But again, with carbohydrates, when they're in water, they typically prefer to form these ring structures, where the number 1 carbon and the number 4 carbon form a ring by forming this hemiacetal group. And so in this form, it's also called the furanose form. But this is how ribose looks when it's part of a nucleotide. The 4 carbons and the 1 oxygen that form this 5-membered ring are almost all in one plane, so they don't quite form a single plane. Typically, the ribose molecule, the 5 atoms that make up that plane, are 4 of them usually are in one plane, and 1 is above or below the plane of the other 4. And so there are a couple different configurations there. Usually either the 2' or the 3' carbon is above or below the plane. If it's above the plane, it's called above with respect to the 5' carbon. So the 5' carbon is the one the one we're in reference to. So if it's above the plane with respect to the 5' carbon, then we call it the endo configuration. If it's below, it's in the exo configuration. So it's a ring, but it's not quite a flat plane, and there's some flexibility there. And then the base is what makes each nucleotide different. So every nucleotide has the same ribose. So it's either ribose or deoxyribose, but then the base is what makes each nucleotide different. So again, to draw an analogy with amino acids, remember that each amino acid has a unique or has a different side group or residue associated with it. So the base is the corresponding group with nucleotides. Each nucleotide can have a different base attached, and there are basically two types of bases that can form nucleotides. The pyrimidines are six-membered rings that look like this, with two nitrogen atoms part of at the 1 and 3 position, and then the purines form two rings. So one six-membered ring again. Here the nitrogens at positions 1 and 3, and then nitrogens also at positions 7 and 9. By the way, this is also why the atoms in the ribose sugar are given the designation prime. So the reason we call the carbon that the base is attached to 1 prime, and then we go 2 prime, 3 prime, 4 prime, and so on, with the 5 prime carbon sticking up, is that the atoms in the base get numbered first. So with pyrimidines, there's 6 atoms in that ring, and starting with this nitrogen, we call them 1, 2, 3, 4, 5, 6 in clockwise order. And then with the purines, they have their own numbering system. Now, the two purines that typically form nucleotides are these two. They're called adenine and guanine. And adenine, you can see, has again, they both have that same 2-ring structure. By the way, if you have trouble remembering which ones are the purines or which ones are the pyrimidines, the way I learned it is that if you because the purines kind of have this double ring structure, if you kind of use your imagination a little bit, you can think of the purines as having almost like a heart shape, with a bond down the middle, that double bond down the middle. So the way I learned it is that purines are pure of heart, and that is how you can remember which one is which one's the pyrimidine, which one's the purine. It's silly, but it works. So adenine and guanine are here. Again, you can see they have very similar structures. The 5-membered ring for each one is the same. The only really place where they differ is that on the number 2 and the number 4 carbons have different functional groups. So for adenine, the number 2 carbon has just a hydrogen, and the number 4 carbon has an amino group. Whereas for guanine, the number 2 carbon has this amino group, and the number 4 carbon has a carbonyl group instead. Meanwhile, the pyrimidines, there are actually at least 3 different pyrimidines that can form, that can be part of nucleotides. So those are cytosine, thymine, and uracil. So again, these are all 6-membered rings. Cytosine, you can see at the number 2 and 4 carbons again, you have a carbonyl group with number 2 and an amino group at the number 4. Thymine is very, very similar, except that instead of an amino group at the number 4, there's another carbonyl. That also means that the 3 carbon has a hydrogen attached. Meanwhile, uracil, and then thymine also has this extra methyl group on the number 5 carbon, whereas uracil, in fact, looks very, very similar to thymine, except that there is no methyl group. So, in fact, uracil is or thymine is really just methylated uracil. And the other difference between thymine and uracil is that thymine is only found as a part of DNA. Uracil is only found in nucleotides that are part of RNA. So the ribonucleotides then there are 4 of them, and they're named after the base that they contain. So a ribonucleotide that contains adenine as a base is usually called the nucleoside anyway. The base plus the ribose is called adenosine. And so if you have a nucleotide of adenosine, that's adenosine monophosphate, and typically the phosphate, again, if it's attached to the 5' carbon, then we call it adenosine 5' monophosphate. But usually we just say adenosine monophosphate, or AMP. Technically, this would be called adenylate. So adenosine monophosphate, adenosine 5' monophosphate would be called adenylate. but again, most people just say adenosine monophosphate or AMP. And then if you have a ribonucleotide with guanine as the base, we call that guanosine. And then a nucleotide of guanosine, we call it guanosine monophosphate. And then guanylate would be the technical name for that one. Uracil, if uracil is the base, when it's attached to the ribose, we call it uridine. And then uridine monophosphate would be full nucleotide. And then finally, a ribonucleotide with cytosine attached, we call cytidine. And then the full nucleotide is cytidine monophosphate. A lot of times these are just abbreviated with the first letters of the bases. So adenosine is just A, and then adenosine monophosphate is just AMP. Guanosine is G, guanosine monophosphate is GMP, and so on. And U, UMP, C, CMP, and so on. Importantly, the structure of the nucleotide is always the same. So you always have the ribose, or the base attached at the 1' carbon of the ribose, and the ribose attaches to the same relative position on the bases. So on pyrimidines, the ribose is always attached to the number 1 nitrogen of the ring, and for purines, it's always attached to the number 9 nitrogen of the 5-membered ring there. So that's the general structure of the ribonucleotides. The deoxyribonucleotides are, of course, very similar, with the exception that on the 2' carbon here, the 2' carbon, there's no oxygen there, so that's why we call it deoxy. So if adenine is the base attached to your deoxyribose, then that would be deoxyadenosine. And then with a phosphate attached, we call it deoxyadenosine 5' monophosphate, or just DAMP. For guanine, it becomes deoxyguanosine. For cytosine, it becomes deoxycytidine. And again, thymine is a base that's only found associated with the deoxyribonucleotides. So deoxythymidine would be nucleoside form of thymidine or thymine, and so then deoxythymidine monophosphate would be the full nucleotide, or deoxythymidylate. So in other words, there is no such thing as just thymidine. I mean, you can have thymidine, of course, if you had a thymine attached to a regular ribose, not deoxyribose, then it would just be thymidine. But in reality, in most cells, most of the time, thymine is only attached to deoxyribose, so it's always called deoxy. The technical name should be deoxythymidine. And then again, we put this little lowercase d to reflect the fact that it's a deoxyribose. So adenine is A, and then deoxyadenosine we just say is little d with a big A, and deoxyadenosine monophosphate is little d AMP. And then, like I said, you can have other bases attached to the ribose. So there's lots of other potential nucleotides you can make. Any version of a pyrimidine or a purine attached to a ribose will give you some nucleotide. So, for example, most of the nucleotides have methylated forms. So adding a methyl group creates a different nucleotide. This is methylcytidine. This is methyladenosine with the methyl group here. You have methylguanosine at this position, and then in this case, this is hydroxymethylcytidine. So there's a methanol group attached to it. Some of these modified nucleotides are important. So methylcytidine, for example, and methylguanosine, those are show up in DNA and RNA sometimes. So sometimes methylating DNA is an important way to change how it interacts with proteins, for example. And then there are modified versions of nucleotides that show up in other forms. So these two molecules, inosine and pseudouridine, which are inosine is a slightly modified form of adenosine with basically a carbonyl oxygen instead of an amino group up here, and then pseudouridine is a form of uridine where the ribose is attached to the number 5 carbon instead of this number 1 nitrogen down here. And these are important because they are actually found in certain forms of RNA. So transfer RNAs and ribosomal RNAs, which we'll talk about what those are later, can contain these modified nucleotides. And then this is another example. This is another methylated version, methylguanosine. And you can even have sulfur groups attached. So this is thiouridine down here. Now, you don't see, for example, anywhere on here so far, at least a modified version of thymidine or thymine, because in fact, if you had or for example, a methylated uracil, because again, a methylated uracil would just be a thymidine. Now, the phosphate group again is what makes the nucleoside into a nucleotide. The typical form of a nucleotide has the phosphate group attached to the 5' carbon, but there are other modified nucleotides where the phosphate can be attached to other carbons. So this, for example, is a 2' monophosphate version of adenosine, where the phosphate is just attached to the 2' instead of the 5'. And you can have a 3' monophosphate down here. You can even have this structure where you have, because there's 2 hydroxyl groups there, one on the 2', one on the 3', both of them can be attached to the same phosphate. You can form essentially a 5-membered ring like this, and this is called a cyclic monophosphate. And these cyclic monophosphates, so adenosine cyclic monophosphate, or cyclic adenosine monophosphate, I should say, is an important molecule for intracellular signaling, which we'll talk about later. All right, so next time we will talk about, that's basically all we need to say for now about individual nucleotides. But next we will talk about nucleic acid and kind of how we know, in fact, that nucleic acids are the genetic material.